Hi everyone, this is Mr. Maneno from Gainesville Middle School, and today I'm in my kitchen and I want to do a short video about how to use a microscope. Microscopes are one of the hallmarks of 7th grade science when we learn about cells, and so obviously this is not ideal. We'll try to do this if and when we get back to school. I'll definitely try to get microscopes in your hands, or we have a lot of things we can look at. So I just want to go over a quick tutorial, a little bit about our microscopes and how they work, and then try to look at a few slides. And then what I'll try to do is, once I'm done videoing, I'll try to get things focused and use my phone. A lot of times if you put a phone or a camera on top of the lens, you can get a pretty good picture of what you're supposed to see. So I'll try to show you that real quickly. So microscopes come in different varieties. This is a compound light microscope. This is also a light microscope. This one is just one that I bought for myself probably 15 years ago. And the way you figure out the power of the microscope is you look at the eyepiece. And again, I know this is kind of hard to see. But this one says 5x on it. That's the eyepiece. And then the lens, and again, I know this is hard to see, but this lens has a power of 10 on it. So what you do is you multiply the eyepiece times the lens. So this is 5, this is 10. So this has a magnification power of 50. 5 here, 10 here, 5 times 10 is 50. And you can kind of see this one works by just this tube here collects the light, so the light shines up through the slide that way. Let me move this one aside. This is nice because it's portable. You don't have to plug it in, so you can take it outside and look at things. That's really the benefit of that one. Typically, our microscopes in school are like this. They're called compound light microscopes. Light, because there's a light. Compound just means more than one. So like, you may have heard of like a compound fracture. When someone gets a break in their arm or leg, they've broken multiple places, that would be a compound fracture. So here, compound, more than one lens. I can spin this around and use different powers. Now, if you had to guess, there's a short lens, there's a medium length, and there's a medium one and a longer one. If you had to guess which is the more powerful one, what would you probably guess? You'd probably guess the long one, and you'd of course be right. So what we do with the light microscope is we always start on low power. So I want to go through just a couple of parts real fast with you and then show you how to use this real quickly to look at a few things. So most of the parts kind of make sense. Eyepiece, part you look through. Objective lenses or lenses, this is where the magnification comes from. This is called a revolving nose piece because it spins around and you can change the power. The stage is where the slide sits, right? So the stage in a play would be where the actors are. Here the stage is where the slide sits. And then what happens is, I know it's hard to see, this little is called a diaphragm. It's a little disc that adjusts the amount of light that shines up through it, so I can adjust that. And of course we have some adjustment knobs. This is a coarse adjustment knob. You can see if I turn this, the entire stage moves up and down. And this is a fine adjustment knob. It also moves the stage, but does it very slowly. So in general, the way we use a microscope is, let me plug it in. Plug it in, turn on the light. Then we take a slide with something on it, and we put it on the stage. We start on low power, and we just simply look, and we adjust this up and down until we bring the image into focus, whatever it is we may be looking at. Now I want to show you how to make a wet mount slide really quickly. So I'm going to try to make a couple real quick slides to show you. First thing I'm going to try to look at is cork. So this is from a bulletin board. And cork comes from the inside of a tree. So what do you think? Cells or no cells? Yeah, it's from a tree, so you're definitely going to see cells. Now, what part of the cell are we going to see? I mean, is there going to be living parts inside of this? No. What we're going to see left over in a piece of cork is the cell wall. Same thing is left in a piece of wood, right? If I have this old cutting board here. Right, what's left there are just the cell walls. The wood is dried out. So when they cut down trees to use for lumber and things, they put them to a kiln and it dries them out, or you can air dry it, and eventually all the living material, the cytoplasm, will dry out, and what's left are the cell walls, that non-living layer of cellulose that surrounds a plant cell. So the trick with seeing cells with a light microscope is to get a thin piece. So really, if I break off a little piece of this cork and I take a look at it, I'm not going to see great cells unless I look at the edge where it's torn, because that's where I can get a thin enough piece. And what I'll see, it almost looks like little kernels of corn or something like that, where I can bring it into focus. Now again, why am I showing you cork? I'm showing you cork because a scientist named Robert Hooke, back in the 1600s, an English scientist, he wanted something interesting to show some of his scientist friends. And so he had an early microscope, and he looked at some cork, and he thought that they looked like little rooms. And the word cell in Latin means little rooms. They reminded him of, today we think jail, prison cells. They reminded him of monk cells, so little rooms where monks would stay to pray. They kind of looked like that to him, so he gave them the name cells. So the reason I always like to start with looking at cork is it's the earliest thing a person looked at, Robert Hooke, who came up with the name cells, looked at a piece of cork just like this. And it certainly wasn't on a slide or something that he purchased from a company. It was just a piece of cork. So I always like for students to be able to see that, because that's how cells got their name, from this scientist Robert Hooke back in the 1600s. So that's the first thing I'll try to photograph if I can do it. The trick with seeing cells is to get a thin piece. If I just took a leaf off of this plant, the leaf would be too thick and I really couldn't see the cells. Now I can try to take a pair of tweezers and kind of peel a very thin section off of the leaf. And if I get a thin enough piece, then I can put it on a slide and take a look. So what I have here is just a blank slide. I'm going to put a thin piece of the leaf on there. And I'm going to make what we call a wet mount slide. So a wet mount slide just has a little drop of water to hold everything flat. So I'm going to put a little drop of water on top of my leaf. And then I'm going to use this little cover called a cover slip. It's just a little piece of plastic. And it really does two things. It holds the specimen flat so you can see it, and it protects the lens from hitting the object we're trying to look at. All right, so what I do now is I've got my piece of leaf on there. If I did a good job pulling off a thin piece, I'll be able to see some cells. Oh, I can definitely see them there. They look like little green circles, and again, I'll try to photograph this if I can. Once I see it on low power, then I can spin this around and go to medium power. Now, medium power in this microscope has an eyepiece of 10, and medium power is 10. So 10 times 10 is 100. So now I'm looking at something 100 times bigger than it normally would be. Again, why do I need a cover slip? When I go to high power, the length of that lens is almost going to be right on top of the slide. That's not a design flaw, it's just the way light microscopes are. So that lens, imagine if I just took the leaf and just dragged the lens into it, right? The leaf's going to dry out, it's going to stick to the lens. So a cover slip also protects on high power. And really the only real rule I go over with students is when you're on high power, use only the fine adjustment knob, this lower knob, because that way the stage will move very slowly and you won't risk damaging the lens by turning the coarse adjustment knob. Right? Again, if I go back to medium, this coarse adjustment knob moves the stage very quickly. So imagine on high power, students do this. This is how slides get cracked. They crank that up into it. And you really won't see it that way anyway. Always better to find it on medium, then go to high power and you'll be able to see it. All right, so again, I have a pretty good slide here. I'm going to take this off and try to photograph that a little bit later for you. A couple other things I want to show you. One is onion. Onion's a classic place to see a cell. The reason for that is you can get a really thin piece. So again, not a small piece, but a thin piece. So I'll try to just use my finger here. I'm going to use a pair of tweezers. So what you can do is you can grab an onion, because they're made of layers. Right? Remember the ogre in Shrek? Layers. So same idea. I've got a real thin piece here. I'm going to put that on top of my slide. And again, I'm going to put a little drop of water on top of it. This is actually a little bit of dye or stain, so it'll show up better. And then I'm going to put my cover slip on top. And again, this is called a wet mount slide. I'm going to go back to low power to start. Put my slide on. Turn my coarse adjustment knob to bring that into focus to see the cells. And there they are. I can see them pretty well. Again, I'll try to photograph this just so you can kind of see what I'm seeing, and I'll try my best once we get back to school to show you this as well. But that's a wet mount slide. I'll show you one other thing you can do. Now again, we would not do this in school. We can't do anything with blood or saliva, but because I'm here in my house, I can do this. So what I've got is a toothpick, just not a sharp one, and I'm going to try to look at the cells from the inside of my cheek. So I'm just going to take this and scrape my cheek, not hard, I'm not going to hurt myself or anything. Scrape the inside of my cheek, swipe that on the slide. Again, I can't see anything on here. And put a little drop of this stain on them so they show up a little bit better. Cover slip. And let me see if I've got any cheek cells in there. Yeah, there they are. I can see them. Yeah, they look like little round circles. So again, I'll do my best to try to photograph that to show you. Really, if I don't stain them, cheek cells are going to be translucent. They're really clear. They're hard to see. But if you put a drop of dye on them or stain, then they'll show up and you can actually see them, and you usually see the nucleus. So again, what I'm going to try to do is look at several different things. I've got some prepared slides here. I've got some leaf cells, some nerve cells, some human blood cells, some liver cells. So we'll try to get a pretty good look at some different plant and animal cells just so you can see what they look like. Again, a compound light microscope, some of the parts real fast: eyepiece, objective lens, arm, base, light, stage. Right, most of the names mean what they say. Coarse adjustment knob just moves things very coarsely or very quickly. Fine adjustment knob. And again, the only real rule is on high power, use the fine adjustment knob so we're not cracking any slides. Again, hopefully I have a bunch of different slides and things we can look at when we get back to school. But I just want to give you a real quick look at a compound light microscope. And again, what I'm going to try to do as best I can is use my phone to put on the lens and see if I can photograph some of this, just to show you kind of what some of these cells look like before we really get into the organelles and the parts and that kind of stuff. So from this, Robert Hooke looked at cork cells, came up with the name cells. I'll show you what they look like in just a few minutes. How to make a wet mount slide, thin piece, slide, specimen, cover slip, and a real quick look at the compound light microscope. All right. Thank you, folks.